right, I think we're going to go ahead and get started. I'd like to welcome everybody to the second lecture of the science series that SCCR is giving every 2 months. Our first one was on cardiac anatomy and physiology with basic EKG interpretation, which was in September. The second one is obviously today on biospecimen sampling and clinical trials. And then we will have a third lecture in January on pharmacokinetics and pharmacodynamics. So look out for the email. There'll be another sign-in for that. For Department of Medicine coordinators, the brown bag is on Thursday this week. You should have gotten an email, so we look forward to you meeting us there. Wanted to just also mention that this is for, these classes are for the research professionals, research personnel on these studies. So if there's any topic that you guys want us to present on, and these science series are really supposed to be more pathophysiological and biological in nature. The regulatory classes we will have, we're setting up training for informed consent in January, and then regulatory binder sometime probably February, March. So for non-regulatory things, science series topics, please let us know. We're happy to work on some topics that you guys are more interested in. And I just wanted to briefly introduce our speaker. So this is Sumana Shashidhar. She began her career as a basic research scientist in the biotech industry in 2002. She joined the Department of Blood and Marrow Transplant at Stanford in 2004 in the lab of Dr. Wes Brown. She developed and established animal models of hematopoietic progenitor cells-based therapies to prevent opportunistic infections in the setting of stem cell transplant. Urine Therapeutics, a biotech company which is now conducting clinical trials in human subjects, adopted development of her work using myeloid progenitor cells to counteract neutropenia resulting from radiation and chemotherapeutic pre-transplant conditioning. In 2010, she shifted her focus to clinical research and became a study coordinator in BMT, working on over 14 industry-sponsored studies. She joined the SCCR team in 2015 and helped support and promote clinical research by faculty in the Department of Medicine and their collaborators across the School of Medicine and Stanford University. So welcome and thank you.
Thanks, Kira. Hi, everybody. Welcome to this class on biospecimen sampling in clinical trials. Well, give me a second to get this plugged in. Right. So the goal for teaching this class is for you to walk away with a broad understanding of why we collect biospecimens in clinical trials, what kind of specimens we collect in clinical trials, how we collect them, how we store them, and what that has as an effect on the data that we seek to obtain from them at the end. Now, a very broad definition of biospecimen is it's a biological specimen that you collect from humans, animals, plants, which are stored in biorepositories or biobanks, and you carry out laboratory research on them. Now, for the rest of the talk, every time I say biospecimen, I'm going to be referring to specimens you get from humans. Purpose of collection. The most important purpose is that when you collect a biospecimen at any point during the trial, whether it's based on a predefined schedule of events that's defined by a protocol, or whether it's based on a clinical event that happens to a participant in the study, what this does, a collection of a biospecimen, is provides a biological snapshot of what is going on with that participant physiologically at that point in time. And a biospecimen is collected as a representative of what is going on and as a representative of any other sample you would collect from that source. And of course, it's collected so it can yield important information about what is going on and in answering the research question. Let me give a little example. Let's say there's a vaccine being developed against a virus that's causing rash X. Now, the vaccine is made up of viral components, and it's a placebo-controlled trial. Half your participants get the vaccine, half the participants get placebo. You don't know who's getting what. And some of your participants get rash X. Now, when a person walks in with a rash, you can't collect every single rash sample on their body. What you do instead is collect a representative, and you hope that the representative you collect from them will give you the same information as any other rash you would collect on their body. And when you collect the rash, you can do things like analyze it and see, how did this rash come about? Is this rash being caused by the wild-type virus, meaning did they just acquire it in the community? Or did the virus in the vaccine cause it, which can answer questions like safety and efficacy. And these principles can be applied to pretty much any trial that you're conducting. Now, irrespective of the kind of trial, most clinical trials have a very general life cycle that they follow. It all starts with a participant. They sign informed consent, agree to be in your trial. You have a collection protocol, whether it's based on a protocol schedule of events, whether it's based on a clinical event. And based on that, you collect biospecimens. Now, there are a variety of biospecimens that you can collect, and I will go into that in greater detail as I proceed through the talk. Once you've gotten the biospecimens, you process them, you biobank them, and then begins the laborious work of research and analysis. And usually, this gives rise to more questions. I guess that's why it's called research and not just search. And so when these questions come up, you often have more clinical trials, phase 1, phase 2, phase 3, where you're having this clinical trial. You're expanding your sample size by having more participants, by including a more diverse group of participants to see if your answers that you're getting from or the results you're getting from your previous trials bear out in future trials. And usually the gold standard is you want to get FDA approval if you're a drug or a device, and you want to go into the market and be of commercial use. Or if it's an observational trial, you want to get data that you can interpret in order to build better biomics or proteomics, or get more answers about how you can personalize medicine and make better drugs for people. Now, these are the biospecimens. There's a small list of biospecimens that people collect as a matter of course in clinical trials. Now, I just want an informal show of hands. If you ever collected any one of these biospecimens as I read them off, or you plan to collect them, or you are collecting them currently, just show your hands. I'm just curious to see how many people collect what. How many people collect blood? Okay. So urine, stool, tissue, any kind of tissue, saliva, swabs, cerebrospinal fluid, 2 hands, bronchoalveolar lavage, 3 hands. Anybody doing reproductive tissue, ovarian tissue, semen, vitreous fluid, that's the fluid that surrounds the eye, and synovial fluid, that's the fluid that surrounds the joints. So as you can see, there's a wide variety of specimens you can collect, and I sent out a survey when we were proposing this class, and blood was the winner. So for the rest of this talk, I am going to focus on blood. I will get to it. It is there in the future slides. And that's the other thing. Stop me at any time for questions. If that information is in forthcoming slides, I will indicate that. So blood, very favored biospecimen. And there is both a practical and a biological reason for that. It's fairly easily available from participants. You get it as a process of either venipuncture, where you do a needle stick, or you can get it from line access in people who have indwelling catheters. Now, the biological reason why blood is so important is because it circulates all over the body, touches upon pretty much every major organ system, and when it does that, it also samples their milieu and brings back important information which you can then draw, and you can study, and you can analyze, and you can answer your questions. It's also a very complex fluid. It's a mix of different components, and people have figured out ways to separate each and every one of these components and be able to study it in tests and assays and be able to get at the answer they're looking to find. So let's take a look at the components of whole blood. Usually when people talk about that, they are referring to plasma, which is the fluid component of blood, and the formed elements or the cellular components of blood, which are the red blood cells, the platelets, and the white blood cells. We use centrifugation to separate out these blood components. If you draw a vial of blood and place it on a counter, at some point you see a separation between the formed elements, that's the cellular elements, and the liquid portion. But when you do that, it's sitting at room temperature, a lot of factors die off, you might not get the analyte or the fraction you're seeking to assay. And more importantly, as busy study coordinators, you don't have the time to let a vial of blood sitting around affecting that separation just based on gravity. So you use centrifugal force, and what that does is it separates out the liquid fraction of the blood from the cells. And you can do it in centrifuges using tubes of blood, and blood banks, which do it on a much larger scale, are able to do this with the bags that they collect blood into. Now, when people talk about components of whole blood, these are the common ones. And then people start lobbing terms like, say, serum. And a lot of people sometimes use serum and plasma interchangeably, but actually they're not the same. People will talk about Buffy Coat and PBMCs, and they will talk about them interchangeably, but they are not the same. So now let's look at what these mean. So the function of whole blood is transport. It transports gases. It transports nutrients. It carts away waste products from the tissues it goes to. It carries hormones, enzymes, electrolytes, metabolites, whatever you can think of. It also helps in temperature regulation. Now, depending on whether it's a hot day or a cold day, if it's a hot day, your veins actually move closer to the surface of your body to affect evaporation, so you can cool down by sweating. And on a cold day, they actually go inside, closer to the inside of your body, so they can conserve the warmth. And this is how they help in temperature regulation. They help in immune defense. They contain the cells of the immune system, and they also help in the pH balance of the blood based on the electrolytes and certain proteins that are in blood. Now let's look at these two terms that people sometimes use interchangeably, plasma and serum. Now plasma is a little over half of the total volume of blood. It's mostly water, but it also contains the important electrolytes and antibodies, metabolites, hormones, enzymes, blood plasma proteins, and these are fibrinogen, albumin, and globulin. And its function is to act as a transport medium for all these things, along with blood cells. So what is serum? Serum is very similar to plasma in its composition, except for one important factor, and that is fibrinogen. Serum does not contain fibrinogen, and it has all of the characteristics of plasma except for that. So plasma minus fibrinogen is serum. It's mainly used for chemistry tests. Now you might wonder why, if they're so similar, you can't use them interchangeably. That's actually because serum contains a more concentrated amount of antibodies, of antigens. It also contains all the things, the analytes that you're looking to assay in chemistry in a more concentrated way. Whereas if you look at the same analytes in plasma, plasma draws out water from the cells and causes this dilutory effect. So you don't have as much of a sensitivity when you're actually trying to study something. So based on the kind of test you want to run, based on the kind of thing you want to see, you have to make a determination about whether you want to look at plasma or you want to look at serum. RBCs, they're the next biggest portion of the blood. They're about 44% of total blood. These are called erythrocytes. They don't have a nucleus, and they contain the protein hemoglobin, which also gives the red blood cells its color. Their function is gas exchange, O2 from the lungs to the tissues, and carbon dioxide from the tissues back to the lungs. I have a rather embarrassing anecdote. When I was taking a hematology class, it was at 7:30 in the morning in San Jose State. We were given a vial of blood, asked to make a blood smear and write a report. The next week, our instructor told us that that was the best grading of scientific fiction he had ever done. You see, all the cells we had looked at were oval in shape, whereas mature red blood cells are sort of spherical with a depression in the middle. And all of the cells that we had seen in that smear were nucleated. And my creative answer was, it's an erythrocytic precursor blood cancer. my lab mate wrote down dead man walking. The correct answer was that it was non-mammalian blood. It was chicken blood. So some lessons stick. Now let's take a look at platelets. Platelets are not actually cells. They are fragments of bigger cells called megakaryocytes, and they're formed by budding. And their main function is to act as the band-aid of the body. They facilitate blood clotting. Anytime there's an injury to a blood vessel, they rush to that spot and form a clot and prevent loss of bodily fluid. They don't always travel around, they don't always travel around looking like they're amped up on steroids. This is only when they're activated. For the most part, they circulate in our blood vessels looking like this, except when they see injury or certain endotoxins in which they get activated and form these web-like things, which then ends up coagulating and coalescing to form a clot. The white blood cells are just 1% of the total blood, and they carry out all of the immune defense of the body. So for me personally, these are the most important blood, these are the most important types of cells in our body. It's not true you can't live without red blood cells and platelets either, but they're divided into two large subgroups. Unlike the RBCs and the platelets, which are fairly homogeneous group of cells, these are divided into granulocytes, and granulocytes are further divided into neutrophils, eosinophils, and basophils. Neutrophils are the vacuums, basically they're the hoover of the blood vessels. They protect us against bacterial invaders, against fungal invaders. They carry out something called phagocytosis, in which they actually eat the invader and clear them from our bloodstream. Eosinophils mostly protect us from parasitic infections, helminthic infections. Basophils are involved in mediating allergic reactions. And we have a second group of cells, which are the peripheral blood mononuclear cells, or the PBMCs. And these are comprised of lymphocytes and monocytes. Again, lymphocytes are further subdivided into B cells, which make antibodies, and T cells, which are involved in presenting antigen to elicit an antibody response, which are also involved in mediating viral attacks. And they're also responsible for controlling cancerous cells in our body. And monocytes are also kind of a vacuum system. They tend to carry out a lot of phagocytosis. They clear the cells of debris. They also engulf bacteria. They also engulf antigens that happen to come across in the bloodstream. These monocytes can actually move from the blood vessel into the tissue. And when they do that, they're called macrophages. And if they move to the liver, they're called the Kupffer cells. Buffy Coat and PBMCs. Buffy Coat mainly refers to the white blood cells that are in the blood. So when you take a vial of anticoagulated blood and spin it down, you get these two fractions, liquid and a pellet. But then when you look at the interface of the pellet and the liquid, you see a tan coat, a buff-colored coat, which is actually how it got its name, Buffy Coat. And PBMCs are a more purified version of that Buffy Coat. Usually people use phycol, which is a polysaccharide, and its density sort of falls in between that of PBMCs and granulocytes. So when you take a Buffy Coat and spin it along with phycol, you're able to affect this separation between just the PBMCs, so you can get the lymphocytes and the monocytes that you want to study in immunogenic assays, and able to separate it out from the granulocytes, which you don't want in your assays as they might interfere with it. Any questions at this point? Okay, so now that we know what the components of blood are and what their functions are, let's take a look at the additives in the tubes in which we collect blood for collecting biospecimens and how they work. So let me make a small segue into how a clot is formed, and this is relevant to the additives that we use. So usually when there's an injury to the blood vessel wall, you have platelets that come in and they start forming a seal. And this slide is actually a little incorrect. It says that platelets start to release fibrin, but that's not accurate. What platelets do is they release a protein called thrombin, which then activates the blood clotting protein that's present in the blood, fibrinogen, and that forms fibrin. Either way, a clot is formed. Now, during this clotting process, which is actually a very involved process, it's almost a cascade. It's referred to as the coagulation cascade. There are a number of proteins that almost in a domino waterfall effect get activated, all resulting in the formation of a clot. And calcium is a very important factor in effect making sure that it goes from that initial activation to the actual formation of a clot. And thrombin is another protein that activates the fibrinogen in the blood, which turns into fibrin, which forms a clot. So why is that important? It's because the additives in the tubes you collect blood in work by affecting the coagulation process in some way, either by stopping it, disrupting it, or by promoting it. So, and I'll go into this a little bit more in detail, but there are three categories of additives usually. There are the anticoagulants, which you use when you want to study whole blood or plasma determinations. There are clot activators when you want to study serum determinations. And then there are some special additives, which are usually anticoagulants, but with some extra special properties. So let's take a look at the anticoagulants first. As their names suggest, they prevent the clotting of blood. Now, there's a few kinds of anticoagulants that people use. One is heparin. This was actually, they first isolated heparin from leeches. Anybody in the audience who's had a leech bite? So I lived in India. In the monsoon, it was impossible to walk through the woods or near a bush without having at least one leech attack you. And it's very interesting, when you detach the leech, if you had a similar scrape or a cut next to it, you could see how much faster that stopped bleeding than compared to the leech bite, which would slowly ooze blood for up to 45 minutes after. That's heparin in action. What heparin does is it inactivates thrombin. When it inactivates thrombin, it prevents fibrinogen from turning into fibrin, and that's how it prevents clot formation. It's also a very gentle anticoagulant. It doesn't interfere as much with the assays, but it has one drawback. It's actually pretty expensive. But there are certain special tests that blood banks do. There are special cytogenetic tests, and when that's the case, when the case need is warranted, you would use heparin as an anticoagulant. Citrate is the other popular anticoagulant that you see. It usually comes in the blue top tubes. This is specifically collected for coagulation studies, and that is because citrate binds calcium, which, as we've seen before, is an important factor for coagulation to happen, but it binds it in a reversible fashion. What it does is, when you draw blood to carry out coagulation studies, citrate freezes the process of that coagulation from taking place. And once it's in the laboratory, you can reverse that binding from calcium, and you can then proceed to watch the coagulation continue, and that's how you study coagulation. There's a very popular brand, which is actually almost ubiquitously used when you're using anticoagulants, when you're not collecting them for coagulation tests, and that's EDTA, ethylene diamine tetraacetic acid. It's reasonably cheap. It binds calcium, but a lot more strongly than citrate, and it binds it irreversibly. People usually use it for hematology studies. Then there's one more called potassium oxalate. And the only time I have seen it used is in a binary system, where it's used with another chemical called sodium fluoride, and it's used for glucose studies. So these are some of the tests that anticoagulant tubes are used for. The top tests, the trace elements, toxicology, nutritional determination, chemistries, hormone levels, and protein levels are tests that can be run both with anticoagulants and serums. But then there are some tests that can only be run when you collect blood into anticoagulant tubes. And those are the coagulation tests, the glucose determination tests, CBC or complete blood count, and immunohematology tests. Yeah, sure. Mm-hmm. 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 Yeah, the citrate. Okay. Uh-huh. There actually isn't. And in the blood banks, people often collect it into citrate tubes even when they're running other studies, not just coagulation, but also the CBCs and immunohematologies. So there really isn't. But citrate is a lot more gentle. And I think that since people use EDTA mainly because it, you know that once you've collected it into the EDTA tube, the, the anticoagulant properties of EDTA are a lot longer lasting. So if you're going to be making, if you're going to be carrying out your assays in any finite amount of time, then yeah, I would say use citrate. I don't think it's going to affect. The one thing I would point out is it would be really helpful for you to also talk to the clinical lab people because they actually have a lab directory. And if you type in the kind of test you want, they can provide you with the kind of tube or tubes you can use to carry out these tests. Exactly, but there are also smaller EDTA tubes that you do get. Yeah, I don't think there's the only thing is if you're not carrying out coagulation tests, that's fine. If you're carrying coagulation tests, then I would say use the citrate. Otherwise, I think you can use them interchangeably. Yeah. Clot activators. These are the antitheses of the anticoagulant tubes that we were just talking about. Here, you want to promote clotting in order to obtain serum. And this is mainly used when you want to do lab determinations on serum samples. There are usually, the most of the time when people use clot activator tubes, they are coated with silica powder. The reason is that people found out that if you take blood and put it on glass, the platelets can actually get activated and form a clot just because of the surface of the glass. You see, glass has a negative charge, and platelets can get activated based on just the negative charge because that is not the normal milieu that they encounter in the blood vessel. So anything that is removed from the milieu of the blood vessel and they see as, oh no, this is not right, they will form a clot. And so people have used this. Silica powder is actually pretty cheap too. And so people use this almost exclusively in all of the clot activator tubes that you collect blood into. Some of the tubes also contain a gel. It's called a thixotropic gel. And basically what it means is that it's an inert gel. It does not interfere with the assays that you're choosing to run with it. And when you collect blood into a serum separator tube, there is a gel. You spin the tube down and the gel somehow moves into the middle so that the cellular components and the red blood cells and the clot are separated from the serum, which you can then extract and run in assays. It takes about 30 to 60 minutes for clotting to be affected completely. And if you want a more rapid clot inducer, some people will use thrombin tubes. These are special orange top tubes that I have literally seen only once in the last 11 years. And they can induce clotting in as little as 5 minutes. They're used when emergency situations warrant that serum is collected as fast as possible. They're very small and they're also incredibly expensive. There are some point-of-care systems that are using diatomaceous earth. Diatoms are these little organisms that live in the sea, and their cell wall is made mostly of silica. Silica, again, has a negative charge, and so this earth called sealite is now being used in certain point-of-care systems in order to promote clotting, so you can get these minute amounts of serum to be able to run point-of-care assays. There are also other tubes called no additive tubes. Yes, I did not leave out a non. It is no additive. That's how they're referred to in the industry, go figure. These are made of glass. They are negatively charged because they're made of glass, and therefore they normally promote clotting of blood. They contain no additives. They do not contain anticoagulants, obviously. They do not contain any added clot activators, and they do not contain serum separation gels. These are used when you want to affect clotting, but you want as little contamination or as little addition from any other added factors in the tubes, and blood banks like to use this kind of tubes in their assays. So these are some of the tests that we can use serum for. And as you'll see, the top part is very similar to the list that I had for the anticoagulant tubes, but there are some certain tests that can be either carried out in one kind of a tube or other. If you're going to be doing a serology test, most labs will ask you to collect it into a clot activator tube. And if you're doing blood bank tests where you don't want any added elements, then you use a no additive tube. Now, I mentioned certain special additives. For glucose determinations, you need something that prevents the glycolysis or the digestion of glucose from happening, so you can have correct determinations of the glucose metabolism. You use something called sodium fluoride. This has anti-glycolytic properties. It binds to this enzyme called enolase, which under normal conditions would convert the glucose into its, would carry out the glucose pathway. And so this is used when you are trying to look at pyruvate, lactate, or other glucose determinations. And this is used with potassium oxalate, which acts as an anticoagulant. There are acid citrate dextrose tubes that are used. Here, the citrate acts as an anticoagulant, and the dextrose actually acts as an RBC nutrient. It protects the RBCs, and this is most exclusively used by blood banks for tissue typing, for HLA typing, and sometimes even for flow cytometry. And then we have these tubes called SPS, or sodium polyethanol sulfonate tubes. These are used when you're looking at identifying an infectious agent in the blood, and these are found in the blood culture tubes. These are anticoagulants. There's this protein complex in our blood called complement, and what it does is it gets rid of any invaders that come in our body. But when you're trying to grow that pathogen, you don't want it to be rid from your bloodstream, and so that's when you would use these tubes. They also prevent the WBCs from phagocytosing these bacteria, and they also help to reduce the action of certain bacteria, so you have enough pathogen left so you can actually culture it and identify what's causing the sepsis. So now that we know the additives, let's take a look at the order of collection and applicable assays. So when I first started out as a study coordinator, I think it was on my second week on the job, I was given a kit with 30 different tubes, and I had a full-out panic attack. Luckily, 20 of those tubes were processing tubes that I just needed to hand off to clinical labs, but then there were 10 tubes left in which I needed to collect blood. I took it over to the inpatient services where the nurse said, "What order would you like me to collect these in?" And I just looked at her blankly. And then she took pity on me and said, "Would you like me to draw it as per our clinical protocol?" I was like, "Yes, please." And so she did that. And I went back to her, and she actually took the time to teach me why they collect tubes in the order that they collect them. So this was me. And there's all these colorful tubes that you encounter in kits. and let's take a look at what they mean. An order of draw is important because you want to avoid cross-contamination between what you're collecting and what you're going to collect. And so a very general order that people follow in the clinic is you first collect blood culture tubes when you're trying to find out the infectious agent. Then you collect coagulation tubes to study coagulation tests. And then you collect tubes that have no additives in it. And then you collect the additive tubes. In this sequence, you first collect the clot activators, and then you collect the anticoagulants. So let's go through that in a little bit more detail. If you're collecting blood in order to culture it for a pathogen, you would collect it into these blood culture tubes, and you can carry out various microbiological determinations based on that. The second in line is actually interesting. I have, again, only come across a royal blue top tube once. This is a very special kind of tube. It's mainly used for trace element analysis, and the reason is the way the glass is made. It's made in a very special way that it has as few contaminants as possible. So when you're trying to look for trace elements, the worst thing would be to have a contaminant in the tube into which you're collecting the blood. These are collected very rarely, and they're also incredibly expensive. So then if you weren't collecting that, and that would probably be the course because they're not part of a routine collection unless clinical need is warranted, you would collect the light blue top, which is the citrate. And these you would collect for coagulation determinations, and these are what they look like. The serum collection tubes come third. And so these are either the red tops, they can be the gold tops, they can be the tiger tops, or they can be the red tops that have no additive in them. Their basic role is to promote clot activation of the blood, by which means you can isolate serum, and you can use this serum for doing serum chemistry, serology, testing for therapeutic blood levels, for infectious disease testing in some cases when you're looking at antigen-antibody reactions. Anticoagulants come fourth, and with heparin. And you have a few flavors of the heparin tubes also, the mint green tops, the dark and the light green tops. The mint green tops actually refer to heparin tubes that have the serum separator gels in them. And so they work in very much the same way that you see the gel working in a serum when you're trying to collect serum, except that here they actually help in separating plasma from the cellular components of the cells. And then you have EDTA. Now the EDTA group is kind of tricky because a lot of the people, when you say EDTA, will say, oh yeah, lavender top. But there's also a pink top. And the reason is, I found out that the color of the caps not only indicates what additive is in the tube, it also indicates what lab that tube is going to be sent to. The pink top, it turns out, is something that is sent to the blood bank. So when people are drawing it, it's an indication for them that this needs to be sent to a blood bank. If there is a tan top, it's an indication that it needs to be sent for a lead determination. And so the lavender tops are the ones that we use for hematology. We use it for genetic testing. We use it for differentials. And we also use it for blood bank testing. There is one cuckoo in the group, in case you haven't noticed, and that is the ESR tube, which is this black top here. It does not contain EDTA, it contains citrate. However, it's collected with this group. And that is because it's used for testing non-specific inflammation. Turns out that when you draw blood and you set it down, the red blood cells will sink down based on a certain rate, and there is a range for that rate. And that's because red blood cells tend to be sticky and they tend to clump together and they're all coming down. But if you have inflammation, there is a separation in these red blood cells, so it takes a much longer time for those red blood cells to separate out. Now, it can tell you there's inflammation. It can't tell you what's causing the inflammation. You would have to carry out other tests to figure out what's affecting that inflammation, but then once you're giving them drugs to counteract that inflammation, you can carry out this testing again to see if the sedimentation rate has improved, which would be again a non-specific marker of improvement. So the sixth and seventh in line are the gray top, which is the binary system of sodium fluoride and potassium oxalate that's used for glucose determinations. And then we have the ACD tubes, the acid citrate dextrose tubes, and which are used for tissue typing, flow cytometry in blood banks, and also for some immunology studies. They either come as a solution A or a solution B. Now they contain the same components, but in different proportions, and that is because that is based on how much blood you're collecting. Solution A is if you're collecting 8.5 mils of blood, the amount of citrate in that tube is titrated to that level to that amount of blood that's collected, and it's 6 mils if it's solution B. So we've looked at all these tubes, we've looked at how you collect them. Now let's take a look at how you would store them. Usually when people talk about storage, there's 3 temperatures they're usually talking about. One is ambient, which is usually storage and even shipping at room temperature. They're talking about refrigerated, which is at 4 degrees, and frozen, which can range anywhere between minus 20 degrees to minus 80 degrees or lower if you're storing in liquid nitrogen. So when you're looking at how you want to store something, you have to look at the stability of what you're trying to study at its required temperature. Depends on how the analyte is being assayed, meaning if you freeze something down, can it be thawed and can you still get the same result as you would get if it was being analyzed as a fresh isolate? And it also depends on how long that particular analyte is stable at that particular temperature. What if there was something that was stable at ambient for, say, 2 days, and then you left it for 1 more day and said, oh, that's okay, let's just run it. I mean, 1 day can't make a difference. Apparently it does. There are people who've studied it in great length, and dependent on how long it's stable, you can actually start to get a decrease in the amount of value because your product degrades with time. The storage, of course, is dependent on the top factor, which is the stability of the component. And it's also dependent on when you store it at a particular temperature and you bring it back to room temperature to analyze it, are your results repeatable? Do you get the same results? Is the sample reliable enough that you can trust the values you get with it after you have stored it and then you're using it as compared to a fresh isolate? And these are all points that you need to think about when you're storing your samples. So serum or plasma, it's stable for about 3 to 5 days at ambient temperature, and it depends on the kind of things you're trying to analyze. Serum can be frozen, plasma can be frozen, and you can use them for certain tests. And usually they're considered to be stable for 2 to 5 years when they're frozen. Whole blood, it's stable for 2 to 3 days at ambient temperature and in a much shorter time for molecular genetic tests. Usually if you're trying to do a PBMC immunogenic assay, you try to ship it overnight, but you can also isolate the PBMCs and freeze them down, which actually extends their shelf life. Sometimes people will freeze a whole blood vial and use it to isolate virus for some studies. As I mentioned, Buffy Coat, which is the WBC fraction of the blood, the white blood fraction of the blood, and PBMCs can be stored frozen, actually indefinitely, and they can be frozen and they can be shipped frozen at -70, and they're used for flow cytometry studies, DNA studies, cell-based assays. RBCs are not that viable. They can be stored refrigerated for about 6 weeks, 42 days, after which they start to degrade. And they're mainly used for transfusion. Platelets have a much shorter shelf life. They can only be stored at room temperature for about 5 to 7 days. And again, this is why people don't store platelets and they prefer to isolate them fresh and transfuse them fresh. long-term storage. Now, there are some blood components that, like plasma or serum or WBCs, that can be frozen down, and you can freeze them at -70 or lower. Each lab, each PI has, each study, each sponsor has to make that determination. How do you want to biobank it? Do you want to keep it in a freezer, or do you want to keep it in liquid nitrogen? And how long-term is long-term? It actually varies based on who you ask and what you're storing. So GLP guidelines say that you can store plasma and serum for about 2 to 5 years, and people in the lab would agree with you. And it's the same thing with frozen white blood cells. If you have stored them appropriately, you can store them for many, many years. There are some basic rules you can follow for long-term storage of samples, and this applies not just to blood, but also to any other biospecimen you would collect. One thing is to avoid repeated cycles of freeze-thaw. We had a lab next door that ordered a brand new freezer and stored all their samples in it, and then a few months later when they opened it, they could not get anything out of these tubes. Turned out they had forgotten to turn the switch off that says automatic defrost, so that freezer had gone through multiple cycles of freezing and thawing, and their samples were worthless at that point. So in order to avoid that, I would say aliquot, aliquot, aliquot. If you get a sample, make as many aliquots of it as you can. It's a good practice. Make sure that you're monitoring the temperature at which you're storing your samples. Make sure that the temperature is being maintained. Things are easier these days with technologies where you don't have someone trudging in at 5 a.m. or 6 a.m. in the morning to see if it still says -70 degrees. You have automatic data loggers that can record these temperatures for you and let you know if there are temperature excursions. Store backup samples in backup systems. Don't put all your eggs into one basket. Mechanical things fail. So if you have the option, try to store these samples in different areas, in different locations. And make sure you know what's in that freezer. Make sure you know that you have an inventory of the samples. If you haven't labeled your tubes appropriately, then you don't know what patient was it taken from, what study was it taken from, what date or year was it taken from, or simply what is it? Because when you freeze everything down, it all looks the same at some point. So I am not going to go into too much detail about shipping, and I know you will all thank me for that. I just want to point to you that there is a class that's offered in STARS, and that is the Department of Transportation Shipping Biological Goods or Dry Ice. I would encourage each and every one of you to take it if you haven't already. It is a prerequisite, and it is actually a very informative class in case at any time you have to ship something. This gives you a really good idea, because the last thing you want to do is ship out your precious samples and have them sitting on a dock somewhere because they were shipped improperly, or they don't have a dry ice sticker on it, and they're gone. So I will very briefly touch on other other biospecimens. Yes. Mm-hmm. 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 Things have been made very easy for us with the advent of Vacutainer tubes, because they have a system, a vacuum system, by which they will draw the amount of blood needed and no more and no less, if you have the patience to keep the tube on and actually draw it to the point where the blood stops being collected. For coagulation tests, because citrate is titrated very carefully to the amount of blood that is collected, I would recommend that you fill the tube up. With EDTA, it doesn't matter as much. With EDTA, if you collect a certain amount of blood, again, they have titrated the amount of EDTA in those tubes so that no matter how much you collect, it usually stays anticoagulated. Again, for serum separator tubes, again, thank you, Vacutainer systems. They will collect until you reach that point of collection. For ACD tubes, it becomes very important because they actually have a mark which tells you till what level you need to collect. So look at each individual tube that you're collecting, or having a nurse collect, and make sure that you've filled it at least to that point. Each and every tube you collect has an indicator that this is the minimum amount of blood that you need to collect. And so if you collect that, you're golden. Yes. Yes. Exactly. And there are actually pediatric draw tubes that are available that look like little syringes. And so they are able to draw a certain amount of the amount of blood that is needed, which is a very small volume, which is enough to run all the assays. And so wherever people are doing pediatric research, if you are able to get your hands on those pediatric syringe draw tubes, that would be great. Let's look at urine. So next to blood, urine is something that people collect for safety labs. That's because urine is the byproduct of blood filtration and has a lot of the metabolites and things that you see in blood. People often use urine for routine urinalysis, where they're looking at the color of the urine, the composition of the urine, looking to see if there are certain things in urine that shouldn't be there because that is an indicator of disease. People use urine for pregnancy testing because beta-HCG is secreted into the urine, and you can test and see if a participant is pregnant before you decide to start them on a trial or not. Chemistry testing. And if you have an infection, people also do urine microbiology to see what is causing that urinary tract infection. Usually people recommend that you collect it by clean catch, but often you have patients who are not able to pee on demand, mainly because of renal failure. These people often have indwelling catheters, and so then you can also you can also collect from the collection bag. However, it's always important to label how you have collected the urine because it does make a difference in the analysis, especially for microbiology. Shipping to outside labs. Usually urine is sterile. So if you don't have a preservative tube, and people often use preservatives such as 6-normal-HCl or boric acid, or sometimes they use proprietary preservatives to ship it out, what they would recommend is sterilely transfer it to a sterile tube, cap it tightly, and send it off by overnight shipping. I actually had that happen to me on one of my trials where they forgot to ship me urine transport tubes that had the preservative in it. And I called them in a panic and said, I don't know what to do. She was like, do you have a 50 ml conical? Yes, I do. Put it into that, cap it as tightly as you can, wrap paraffin around it, and you'll be fine. And we were. So, stool. Stool is often collected for microbiological analysis. If people are having diarrhea, you collect stool. You can do a microscopic exam. You can culture it. You can do an enzyme immunoassay to see the kind of pathogen, viral pathogen that is causing the diarrhea. You can do a PCR. You can do a visual examination to see if you have been infected by parasites. And you also can do occult blood analysis of stool. Blood in stool is not normal. And so if you have blood in stool, it's probably an indication of some kind of a damage that's happened, and it can also be an indication of malignancy. People also do enzyme analyses because sometimes people come with diarrhea and turns out they have a metabolic disorder. They have a pancreatitis, and they're not making enough pancreatic digestive enzymes that are able to absorb the proteins, carbohydrates, and fats. And so people will also do enzyme analyses to see if there are metabolic disturbances going on in the body. You can sample it one of 3 ways. The most popular one is the hat. It's a plastic apparatus that looks like an inverted Stetson. Do not put it on your head. And so it's the patient or the participant who takes the hat home, collects it, puts it into any number of these vials that the lab will provide to you, or you can provide to them as part of the kit. You can also collect it from the diaper. And in some patients, you can collect it from rectal tubes. And each of these colored containers that's here has a particular preservative that preserves certain features. Either it's just a sterile medium, or it's a medium like formalin that will preserve, say, parasitic eggs or actual morphology of certain parasites that might have come out in stool. And so then you can send it off to the lab for future analysis. Tissues. This is really popular. Most of us at some point in clinical trials will collect some kind of tissue. These are tissues from internal biopsies of either organs or cancerous cells. And this can be a skin biopsy, it can be a punch biopsy. You can collect hair with the follicle attached, so you can do DNA studies. People in my previous department, bone marrow transplant, often collected bone marrow to carry out immunological analyses and to look for malignancies and so on. So there's a very exhaustive list of tissue. I just mentioned a few. People often do pathology studies. They take the tissue, they fix it in preservatives so it preserves its morphology, and they stain it and look at it. And each cell looks each cell type, each tissue type has a very distinctive phenotype. And trained pathologists can look at these types and say, hey, this is right, this is wrong, this is a malignancy, this is an infection. For example, there's a virus called cytomegalovirus. And when you look at tissue that's infected with cytomegalovirus, you see these giant owl-like inclusions. There are these giant cells with this giant nucleus. It looks like an owl is looking out at you. And trained pathologists can actually pick up one cell among many in a tissue section. People will do genomic analysis after extracting nucleic acids, PCRs, RT-PCRs, sequencing. You can also use the tissue to culture for microorganisms. You can do flow cytometry analyses and look at antigen, antibody, or certain cell comparisons. And you can do other immune studies with tissue. Saliva. Saliva is actually turning out to be a very popular biospecimen that's being collected by a lot of clinical trials. That is because it contains biomarkers that are actually really good indicators of how, say, for example, people are going to respond to a drug or not. There was a particular antiviral drug that I was working with, and they did a pharmacogenomic analysis and found out that in the Asian population, circulating levels of this drug were actually higher because of the presence of a gene. And so these are the kind of analyses that people are increasingly using saliva for. And this is, again, it all ties into this whole thing of personalized medicine, where you're able to say, predict how a certain class of people, or how certain characteristics, genetic characteristics can affect the way you respond to a drug. People often collect genomic DNA and RNA and bank it in clinical trials, and they're usually collected in these kits, wherein you basically have your patient drool into it until it reaches a certain level. And when that happens, it's not just saliva that comes out, it's also cells. And so that's how you're able to get important information. There are a lot of epidemiological studies being done, disease detection, because saliva contains enzymes, it contains markers. In fact, there are a lot of psychological studies that did saliva biomarker studies to study the amount of cortisol, a stress hormone, in certain individuals. And people do metabolite diagnostics and radiation studies, because they can actually study methylation, DNA methylation. When people have received radiation, their saliva has more methylated DNA in it than a normal individual who has not been radiated. And so DNA is collected into the collection, DNA or RNA collection kits. We also collect saliva into sterile tubes and spin it down, so we are able to separate out the liquid portion from the cell pellet. And the liquid portion contains a lot of enzymes and biomarkers that we can use in different assays. Swabs. Swabs are great because they allow you to sample a very specific milieu of the body. There are a number of swabs. You can do topical swabs if people have a skin condition called cellulitis, and you can grow it in the lab and you can determine what kind of a bacteria is causing it. People do nasopharyngeal swabs routinely to look for respiratory infections. They take a swab and basically stick it up your nasal cavity. It feels like someone's giving you a lobotomy, and I know because I had it done recently. And but that's apparently how you know that it's worked. And people will collect that and they'll run a PCR on it and identify the virus that might be causing the respiratory infection. This was one of the main ways that people identified who had H1N1 and who didn't 5 years back when we had that outbreak. Buccal swabs, these are basically oral swabs. You take swabs of your cheek cavities. These are also the kind of swabs you send to the National Marrow Donor Program when you want to register as a donor, and they use the DNA in those buccal cells to test and see what your HLA type is and register you as a potential donor. You have throat swabs. That's what your doctor takes from you when you have strep throat and you can't do very much more than just go, and they have you open up and they swab it, culture it, and they're able to identify the organism because it helps them prescribe the right amount, the correct antibiotic in order to control that infection. Vaginal swabs, these are done not just for infection but also for pathology. So you can scrape a layer of the cells, you can stain them, and you can take a look. For example, the women in the audience who have had pap smears are familiar with it because our cells are taken, they're stained to see with a particular stain to see and if there are any evidence of cervical cancer or not. So you can do tissue typing, microbiology, pathology, and these are usually either shipped at ambient or refrigerated. And for a lot of viral studies, they are shipped in this pink-looking medium, which is called universal transport medium, and that usually keeps the viruses happy and viable until they're able to get to the lab that can test for what they are. Cerebrospinal fluid. Now, this is not a normal collection. Usually this is based on some kind of a clinical evidence of disease. People don't, well, maybe they do, but on a routine basis, people don't go doing lumbar punctures of normal people saying, hey, can we have some CSF? Mainly because it's an invasive process and there's always a risk of infection when something like this is being done. It's usually used to diagnose neurological disorders. And the collection is either via a lumbar puncture where they go into the base of your spine with a needle and draw out the cerebrospinal fluid. Now this is the fluid that cushions your brain and runs along your spine. And because it's also in the area of the brain, it also samples certain information about the brain. And so it can provide important information like, say, malignant cells that are present. If there's an infection, you can either stain the cerebrospinal fluid or culture it and see what's going on with that person. Some people have shunts in their head and the fluid can be drawn out that way. And again, that's usually when there's like a clinical indication. There is also some other methods by which people draw CSF. These are called cisternal and ventricular punctures. They're incredibly rare. They're usually done during a surgical procedure. And then CSF can be frozen for chemistry, immunology. It can be refrigerated for cell counts because CSF does contain cells. You can look for markers of inflammation in it. And for cytology to see if there are any atypical cells present that shouldn't be present. And it's at room temperature for microbiology. You would culture it to find out if there is a meningitis or an encephalitis, what is causing it. Bronchoalveolar lavage. There was somebody in the audience who asked me what it was. Was it Trish? Yeah. This is a very invasive process. It is not done routinely. It's only done when you suspect a lung infection and you want to know exactly what's going on. In this, what they do is they basically put a tube, an endotracheal tube. They take sterile saline and they pump that saline into your lungs. And they lavage it so that it samples the milieu of the inside of your lungs, which nothing would usually see other than blood and, I guess, gases, and they draw it out. And when they draw it out, they can And that's what it looks like. It looks like this. They usually do it if they suspect a malignancy and they can't go into the lung. If they can draw out this fluid, they can conduct pathology on it to see if they see malignant cells. My previous boss was an infectious disease, and she often had people who came to her with aspergillus pneumonia, fungal pneumonia, bacterial pneumonia, and in those cases, they would do a lavage to see what was the infection that was being caused and what was needed to be prescribed to that patient, whether it was an anti-fungal agent or an antibacterial agent. And this is actually, there are a lot of dangers with doing a bronchoalveolar lavage. So if you come across protocols where the sponsor will say, oh yeah, we would like some, yeah, no, it would only happen if it was happening as standard of care. If it was already being medically prescribed by the physicians here, because when you put fluid into the lungs, you also run into the dangers of causing a pneumonia. When somebody is already sick, you don't want to make them sicker just for a study protocol. So that's just my thoughts. So I'm almost at the end of my talk. So I am not going to go into too much greater detail with these 3 biospecimens, but I found them fascinating, so I figured I should at least put up one slide that touched a little bit on them. So there's a lot of research that's being done in reproductive science over artificial ovary construction. Often when ovarian cancer hits, it does not know any age. A lot of young women of potentially childbearing potential lose their ovaries, and therefore perhaps their dreams of having children. And people are looking at constructing artificial ovaries. They take ovarian tissue, they actually capsule it, and they implant it into these people who are now going to receive it. The capsule acts as a barrier to prevent rejection of this ovarian tissue. And so go ahead and PubMed it. There are a few very interesting articles that talk about this stuff. Semen is actually used in anti-aging research because it contains a lot of chemicals. It contains a lot of interesting components that people have been looking at to promote youth, and that's all I'm going to say about it. Vitreous fluid, the fluid that surrounds the eye. There is a really interesting research that's being carried out in Italy where they're using vitreous fluid to more accurately determine the time of death in cadavers. So usually when a person dies, people take core body temperature to determine when this person might have passed away. I think they also take the temperature of the liver, and they also look at physical characteristics like rigor mortis. But these are all estimations, and they have a really wide range. So apparently this is being funded by the Italian crime branch. And so there are certain biomarkers that degrade over time after a person passes away. And that is what they're looking for in this vitreous fluid. So stay tuned. And of course, people also collect vitreous fluid to do proteomic research, vision research. And very recently, I don't know if you read that article that talked about Ebola and its persistence even after a person was deemed to have been cured of it in vitreous fluid, which could actually give rise to a reactivation of the Ebola infection in that person. So, synovial fluid. This is the fluid in your joints. There are a lot of cytological and biochemical tests you can carry out for various conditions of the joint, inflammatory conditions, non-inflammatory conditions. And with that, I completed my talk. Any questions?